welcome to a quick video going over the plans for today. You should have gotten a document that's 2 pages, double space or double-sided, that you're going to be using to finish your phylogenetic trees. Just a quick note that when we go to page 2, the second question, which starts with do you see a particular phenotype or trait, just X that off. We don't need to worry about that question. But the other questions you are going to want to answer using examples from your phylogenetic tree or by finding other examples on the internet. So let's talk about what we're doing today. Ideally, when we look at your phylogenetic trees, and there are some examples I'm going to leave on my desk for you to look at, you should have a phylogenetic tree that does the following things. If we get there, this is loading quite slowly. You can do it. First, you've identified each of your species at the end of your poster, meaning that each species has been identified with the genus species, which is our 2 most specific classifications. So Canis lupus would be for wolf. You should also attempt to age each of your species in terms of how old that particular species, or maybe the broader species like wolves, have been around for. Secondly, when we look at the first page of our handout, you're going to want to take some time and do research on the shared derived characteristics or the shared features or the shared traits of particular classifications. The ones that I would focus on are domain, that would be eukarya or bacteria, kingdom, which might be animalia or plants, phyla, so that would be things like chordata or maybe echinoderms if you're doing sea anemones, class, order, and then finally family. Each of these are broad enough that you should be able to find information about what those particular variations, so for example, if I was looking at class, I might do aves for birds, what characteristics do all birds share, and mammalia. And phyla might be something like chordata. You're just finding the information out that defines each of those orders or classifications. The second thing that you should be doing is defining novel or very specific and new features that make your species that you find now currently distinct and specific. So if I go to my phylogenetic tree example here, I have some shared derived characteristics for chordates. They have a central nervous system, they have pharyngeal gills or lungs, they have an endoskeleton made up of bones, and then all of the individuals I see over here have those traits, a bullfrog, a human, and a wolf. Now, when we get into the next breakup, some are amphibians, they have moist permeable skin. That is not something humans have, but it is something that frogs have. And some are mammals. They have mammary glands and they have fur or hair. That would describe humans and wolves. And then down below here are the novel features that I see compared to other species in that group, or very particular to that species alone. So tool making language, a prefrontal cortex in the brain that's very developed. When you are finished that, you should be able to then move on to the analysis. So let's talk about the analysis questions. The first question is going to be looking at a group of organisms that have one common trait that they got from their ancestor that shows very broad or very large differences between those current species. So, for example, I might take the chordates and look at how lungs or gills have changed over time between wolves, humans, and frogs. That is a pretty broad characteristic, a pretty broad trait that might be very different amongst a variety of species. So you're trying to find traits that are common amongst many of your species, but they all look really different, or they all have very similar or very different functions. The second thing that you're going to answer is, do you see traits or phenotypes that are really conserved? Meaning that they haven't changed a lot, and if you look at a variety of species, they all look the same. Does this mean that that trait does a particularly good job of adapting or doing things in a variety of environments? The next 3 examples are really specific, and you might find them, or you might have to Google and get examples outside of your phylogenetic tree. When we talk about divergent evolution or anatomical homology, that means that two related species have taken a shared derived characteristic, such as a tail, and they've radically altered the function and structure of that trait, leading it to have very different phenotypes. For example, there might be a tail in a species of monkey that allows them to latch on and grab onto things for movement in the trees. Well, a wolf might have a tail that is a very demonstrative of symbolizing emotions and characteristics and behaviors. Even more radically, a bat's wings and a whale's flippers are effectively the same structure if you look at the bones. So again, do you see examples of divergent evolution in your phylogenetic tree when you look at your current species? Alternatively, you might find examples of convergent evolution where two unrelated species have adapted or evolved traits that appear similar, function similar, but most importantly, they didn't get it from a common ancestor, which means that they both derived these traits or characteristics separately and independently of each other. For example, some snakes are poisonous, some spiders are poisonous, some species of platypus are poisonous. Poison is not a common ancestral trait, but rather something that has evolved independently over time in a variety of species. And then the last example you can think of are vestigial organs. These are organs in species. It doesn't have to be an organ. It could be a trait, a phenotype, a particular characteristic that has been lost or particularly reduced and minimized over time, meaning that they are really unlikely to be seen or important. Human tailbones are a really good example. Secondly, you might find examples where an ancestral trait, such as a nose, has become radically different compared to most of the current species in that group. For example, the elephants and the other proboscideae have really large noses, and no other mammal has large noses for that phenotype. And then lastly, when you think about species, do you see evidence when you look at your phylogenetic tree that most of evolution that created most of the important features for life to occur happened a long time ago? Or do you think the most adaptive and specific changes that happened recently are the most important? If you were to take a human and break us down into all of our traits and functions and features, things like a bone and a brain and lungs are really old, and they're really important to survive. But at the same time, humans wouldn't be humans without an opposable thumb, without the ability to speak, the ability to think critically with our brains. Where do you fall on this line? Do you think evolution has created most of the important traits for life a long time ago, and we see them in most species, or is it really the recent evolution? The recent evolution are going to be the novel features that are only found in your species and no other species. Let me know if you have questions. We can talk briefly about this work on Thursday when I come back. And I hope this video has been helpful. Thank you.